Life operates by the spatial orchestration of proteins. At Pixelgen, we have developed molecular pixelation that analyzes this in a completely new way. Previously, it's been studied with microscopy, which has limitations of light and taking pictures. That means you can't measure as many things as you would like and as many samples as you would like. But with molecular pixelation, you can measure hundreds of proteins simultaneously and obtain their relative location on these cells, which will be very useful for the emerging field of spatial proteomics.
When I met Simon first time and he told me about this idea, as a scientist myself, I know advanced science is on the basis of seeing things. And I have been always caught up on the idea of counting things with DNA. He told me like we could recompose this puzzle of millions of pieces of DNA, and I was immediately caught. I thought like we need to do that.
There are, of course, a lot of challenges in developing a new technology, and I think breaking down each problem into smaller pieces and then trying to design well-controlled experiments to understand each piece individually has been a successful approach for us.
I'm very much inspired about this interplay between biology and technology, but also innovation and people being able to think outside of the box, making it possible to do things that no one's ever done before and see things that no one's ever seen before.
Imagine a ball as a cell. Around this ball you have hundreds of different proteins that regulates the cell's activity, and their spatial relationship to each other especially governs the activity. First, we bind to each protein we want to detect an antibody carrying a DNA tag, and then we add a DNA pixel that co-localizes a number of these DNA tagged antibodies with each other. Last and third step, we unite the DNA pixels with each other, forming a PCR amplicon that we can amplify and sequence with next generation sequencing. A software algorithm then figures out from all this DNA sequence where the proteins are in relation to each other on a single cell.
This allows us to ask much more interesting questions from the data, not only if a protein is present, but also what other proteins are next to it and how is the distribution of each protein across the cell surface. So this hopefully will enable researchers to gain much more interesting information that's not really possible now using current technologies.
Our hope by making this software and this data open access is to basically crowdsource innovation. There is this idea that by making resources available, the community can feed itself and then advance science at a much faster pace.
Here at Pixelgen, I hope that we will be able to help researchers and scientists to gain better and deeper understanding of cellular processes and cellular activity, and hopefully in the end aid development of new treatments and drugs.
We hope to attract researchers that are really innovative, that want to make breakthroughs at the forefront of industry, but also interacting with academia.
The best part of working with Pixelgen is the people. We are a bunch of talents with different expertise, and from the beginning we all came together with our different strengths to put forward our ideas to develop such a new technology, and we are here at this stage launching our product.
The best part of working at Pixelgen, I would say, it's curiosity. There is so much of it. In my work, it's like being kids again and playing with different stuff, but to build something more important and potentially could have an impact to advance the human health.
I think that the best part about working at Pixelgen is being able to grow together with the company while creating a completely new technology that has the potential of exploding our knowledge of biology.
What I really liked about the founding team is that we each come from different backgrounds and have different sort of set of skills and expertise that complement each other very well. But other than that, I think both Alvaro and Simon always have this positive attitude and has a really good ability to view problems objectively. And together, I think that ensures that we solve problems efficiently.
Our vision for the company is to become a leading provider of tools and reagents for single-cell spatial proteomics by deploying molecular pixelation in many research areas of the life sciences.